Okay, so it's my very pleasure to introduce the first speaker, Charles Pineau. He's research director at IRSET and is also leading the Proteomic facility, which means that he's very much involved in a proteomic analysis. And today he will speak about proteomics analysis of male factor. And thank you very much for coming. Also, he's chairperson, I think, or vice president of the French Society of Human Reproduction. So, please.
Thank you. So first of all, I would like to thank the organizing committee and in particular Amelia and Rita for inviting me to share with you some of our work and what we're doing on, as you call it, the male factor. So just a few words. I've been working for over 30 years in spermatogenesis. I'm a biochemist, and now biochemistry has a new name. It's proteomics because we work large scale, I would say. And we're using these wonderful tools to address problems in how spermatogenesis works, how does it goes wrong, and what are the components we could identify and link to infertility. So just a few words, and I'll focus on a bunch of people. Let's say here, right in front of me, if you're lost, you make desperate signs, and I will try to explain better, okay? Okay, so what is proteomics first? So it's biochemistry. So proteomics is a large-scale study of protein repertoires expressed by a genome of a cell, okay? It's a descriptive, quantitative, and integrative approach in modern biology. But the complexity of the proteome is far beyond how a genome is complex. There's no direct correlation between the messenger RNA and the protein expression levels. And proteins, while they're produced, they encounter major events, and they are post-translational modified. And we know roughly 50 to 60 post-translational modification, and there's probably a large bunch to discover. And in fact, I borrowed this calculation from a friend of mine who is heading the Swiss Institute of Bioinformatics in Geneva, Amos Barak. And if you make the calculation for the human genome, which is roughly 20,000 coding genes, you have the alternative transcript splicing, and 20,000 genes doesn't mean 20,000 mRNAs. So we expect the transcriptome to be roughly between 50 and 100,000 transcripts. And those transcripts now are encoding proteins. But in the meantime, 50 to 100,000 transcript doesn't give birth to 50 to 1,000 protein. Okay, that's the post-translational modification I was talking about. So on a, I would say, rather optimistic guess, we can expect 20,000 genes to encode for 5 million proteins, not speaking about the new things in the world, which is the gut microbiome. And the microbiomes, bacteria in your gut, for example, are producing proteins, and some of them are very useful for your health. So if we add this big and major event here, we may come up to 10 million proteins for 20,000 genes, which is a lot of things. So basically, when you're doing proteomics, you're playing with very fancy tool, expensive tool. These are mass spectrometers, that are machines that were used for basically chemists and physicists. Each of these instruments is roughly €1 million, negotiated price. And, but we can do marvelous things, but it's very operator dependent. So you need really a knowledge on this instrument. And there's a new bunch of people coming in biology now. These are analytical chemists because they know how to master these instruments and take information out of it. So basically, what we do, in fact, we compare samples between each other, one condition to another, one cell to another. There's no limitation to your imagination. And we have to extract the proteins, so using classical biochemistry mixtures. We have to pre-fractionate the sample onto gels, for example, because you can imagine an extract from a cell or an organ is very complex. We're talking about 1,100 proteins here. And to be analyzed by mass spectrometers, the proteins have to be digested by an enzyme, which is classically trypsin, and you get some digestion peptides that you inject into the machines, and we get mass spectra, okay? So to simplify, each of these peaks is a peptide, okay? I won't go into details because it's too difficult, even sometimes for me. I'm not a physical chemist, you know. And you can do what we call MS or MS analysis, meaning that if you see one peptide of interest, you can fragment it into smaller ions, and you can reconstitute the sequence of the protein, okay? So this is done automatically, and we end up with huge, really huge data sets. Okay, this comes from almost 10 years ago. When you come up with those large lists of protein, okay, it's a list of protein, and so what? So what we're doing here is to highlight some of the proteins that have very differential signal. And for example, this is an example of what we've done many years ago, and this is very classic. I have dozens of proteins like that in my drawer, and each one has a file, and for each protein I would need a PhD student for 3 years. So I have enough in my drawers to work for the lab for the next 50 years, probably. So for one protein here, we had a protein that were highly expressed in late spermatids compared to younger germ cells. So you do in situ hybridization, you see the mRNA, this is rat testis section, is rarely expressed in late spermatids after meiosis, you see, and we develop an antibody, and the antibody gives almost similar signal, a little later expression with concentration in residual bodies. So that was an interesting protein, and then you get to pure chemistry, biochemistry, to see how it works, what's the function of a protein, because when you're a biochemist, the thing you want to know is what is it for? What is this protein doing, in fact? So using this mass spectrometer, you can do plenty of things. We can see that in vivo, the protein is phosphorylated on one specific amino acid. So if you know how to read this whole full spectra, you see the phosphorylation here. In vitro, we were able to phosphorylate the protein, and we discovered it was probably the long-expected substrate for casein kinase 2 in the testis. And this is a phosphorylation pattern. We use very fancy tools, see physicists, and we discovered that the protein is what they call intrinsically disordered protein. We made experiments showing that it had a calcium-binding properties. And finally, we end up with very ancient tools, but still very efficient, using electron microscopy. And we discovered that the protein was associated with the inner membrane of the mitochondria in the late spermatid, so meaning in the sperm. So far, we know that it's a phosphorylated protein. It has calcium-binding properties, and probably the folding events that take place in vivo have something to do with the function. So the next step is to produce a knockout mice and to check what the knockout will do. And so far, we have no knockout, but we don't have phenotype. So we block. We have to discover the stress that makes that protein express itself, so to understand better. So it's a long, long story. So basically, when you're doing proteomics for dissecting testicular protein landscape expression, people do plenty of things. They've studied the development of the testis, cells, difference between different cells in the semiosseous tubule, effect to the hormones or whatever. And so what? So proteomics, this is, as I say, this is biochemistry, but instead of going fishing, you come with larger instruments, you fish everything, and then you need tools to dig out the interesting protein out of this magma. So what I always say, this kind of approach are interesting, but so what? So what? Well, the idea is to look at your data with a different point of view to gain new information. So the idea is to integrate your data with other omics data sets. So this is the trend actually in biochemistry, in proteomics. So this is what we're doing in my lab. Basically, we do proteomics indeed, But at the same time, we do, and we collect also data from other groups that are doing transcriptomics, for example. And we develop custom database. There's a bunch of geeks fooling around in the labs that are pretty good in bioinformatics. So they already developed things like Under the Galaxy world, which is a fantastic world with people developing tools and you can use for your own research and for plenty of things. We do what I call bibliomics, because nothing beats a person sit at your computer and reading the paper. I mean, there's no machine to do it for you. So bibliomics is kind of important in my lab. And using all these tools, well, we can answer plenty of questions about cellular, subcellular repertoires, expression profile, and the various conditions in between cells. A huge world is opening about proteoform, because each protein has probably several forms, and the testis is a factory for proteoform. So we expect thousands of proteins under different forms. Post-translational modification, with some of these probably specific for gonads. And there's also possibility to validate new protein-coding genes, and I will not talk about it today, but we've recently discovered new genes in the germ cell line. Okay, so let me give you an example. I'll give you 3 examples, and I'll try to make it short. Maybe some of you will read general literature, you know, newspaper and things like that. I've heard about what we call the Human Proteome Project. So you've probably heard about the Genome Project. It took almost 20 years to sequence the human genome. And now, in 2010, in Sydney, we launched a Human Proteome Project. So it's a bunch of roughly 2,000 scientists in the world with the aim to characterize each single protein in the human. So if probably the genome has taken 20 years to be sequenced, considering the technology and its state and eventually new developments, we can only expect that 500 years will be the minimum to sequence the whole human proteome. So I'll be dead, and most of you also, probably. So Human Proteome Project, there's an international society called HUPO, Human Proteome Organization. And HUPO has a group of force to map the anti-human proteome in a systematic effort. So there's several pillars that we talk about: antibody, mass spectrometry, and large databases. And we share the proteome into each chromosome, and each country has taken a chromosome. For France, chromosome 14. For Spain, for example, chromosome 8. So I'm the leader in France for chromosome 14, and a good friend of mine is the leader in Spain for chromosome 8, so he's not working in this field. And basically, we meet once a year, and we check what are the protein identified. So far, we've covered 87% of this proteome. But the remaining 2,000-something protein are pretty tough to catch. Okay, and under this Human Proteome Project, there's 2 approaches: a systematic one to characterize each protein after the other, and another one which is more disease-oriented, where for each protein we're supposed to match it to disease, to problem, pathology, so which is quite interesting. So basically, and all of these scientists use this wonderful resource, so most of these will not tell you anything, because this is dedicated to people dealing with mass spectrometers. But this one might be interested for you, Human Protein Atlas. So this is a Swedish initiative. There's roughly 25 leading groups in Sweden organized around what we call the Human Protein Atlas, and their work is to design antibody for each single human protein. So they're roughly 85% are ready now. And if you need an antibody against a protein you're interested in, go and look at the Human Protein Atlas webpage, and they give this antibody for almost nothing. You don't have to pay expensive prices in Santa Cruz and other shitty companies that will provide you with shitty antibodies for some. Human Protein Atlas, they're really validated, and they're high-quality stuffs, working under plenty of conditions. So basically, there's also NextProt, which is in Switzerland. So NextProt is the human database of proteins. And there's an official release each year in January. So at the moment, this is the protein evidence in NextProt. So there's actually, we consider there's 20,230 human protein coding genes, plenty of isoform, over 5 million variants, which is a lot actually. And there's 2,000 something missing protein. I will tell you about the missing protein because it has to deal with sperm. That's quite interesting. And this is the number of identified protein over the recent years. And you see we get to a plateau. That means those remaining ones are really tough to catch, really tough to catch. And at the same time, we have even deeper and stronger guidelines to make sure that you don't identify crap, which can be possible with a mass spectrometer if you don't know how to deal with this. So what about these unseen protein? We don't see them because they're very low abundant. They may be time of stress dependent. They're tissue or cell-specific expression, and then they can be unstable, and they have particular physical properties. And those missing proteins are quite interesting. And the idea was, what about the testis as a ground to look for novel proteins? As you know, most genes contributing to spermatogenesis are under particular selective pressure. There's a high degree of germ cell-specific gene expression. There's plenty of events that are associated with meiosis, as in the female, and haploid germ cells undergo unique phenomena, meaning that these phenomena are supported probably by very specific proteins. So sperm is an ideal tool, ideal ground to look for a myriad of very specific events. And finally, a few years ago, some people in Switzerland and Sweden, they came up with the idea that the production of protein that have been systematically missed might be restricted to very unusual organs, cell types, as for example the testes. Oh, fine. And the largest number of tissue-enriched gene is found in the testes. For most of these people, that was a surprise. Not for us, because it's been known for at least 10 years that the testis has the largest number of specifically expressed genes. And most of these genes are linked to the male germ cell line, and in particular, the germ cell line after meiosis, so haploid germ cells. So no need to get testis and work on it. I suppose there's no donor for me in that room today. So basically, it's easier for us to work on the spermatozoa, which is quite easy to collect. And that's the source to look for interesting protein. And then we came under the spotlight. That was fortunate because we had the objective to understand spermatogenesis and to understand what was the backpack of the sperm when it goes for a long travel with all the proper equipment. And the idea was to prioritize protein that could be potentially crucial in the cell biology of the sperm and correlate this protein expression or misexpression or defects on the protein with male infertility issues. So basically, we say, okay, let's team up with the Swiss people. So we teamed up with the two teams, chromosome 2 in Switzerland, chromosome 14 in France. So we were roughly 80 people working on that. First, we designed a workflow for unambiguous identification of protein using mass spectrometry. We established a first proof of concept with the first bunch of, that was 3 years ago, of missing protein identified in ejaculated sperm. The year after, 2016, we identified almost 250 additional missing proteins. And last year, we were using a targeted approach to look at very discrete one, but we wanted to use the tools to discover these only specifically. So our target was 38, and we succeed in identifying 12 very rare protein in the testis, in sperm, in fact. So these are examples, plenty of examples of immunochemistry validation using this human protein atlas antibody. And then we could see some expression at early stage in spermatogenesis, or staining on acrosome under formation, or in cytoplasmic lobes, or ring-shaped staining, very specific of the head of lead spermatids. And then, well, to highlight those very rare proteins, I'll make a little detour here about a new technology that is appearing in mass spectrometry. And believe me, take my word for granted, in 10 years from now, you'll see this instrument in every single lab in biology. This is what we call a triper-quad, which is garbage name, but with this instrument, what you can do, you don't need anymore an antibody to follow a protein. You get the sequence of your protein of interest, and you get what we call the proteotypic peptides in the sequence. That means those species of the proteins that cannot be shared by another protein. They are unique to that protein. And then you design this peptide through a company, and you will have this peptide with a little label, radioactive label, okay? And you inject the mixture into the sample where you want to measure the endogenous protein, okay? And the technology is called single reaction monitoring. And then you can follow. I will not get into detail with the quadruples in the instrument. You'll get lost anyway. So this is a way, though. You see these awful things that we know how to read. Basically, this is your protein, I mean, the peptides you see here, and this is the label standard, and you see it's exactly the same. So then you can quantify the protein, absolute quantification. You can follow a protein very specifically, with no antibody. And believe me, when you go for a blood test in the lab at the street corner, in 10 years from now, they will use this instrument instead of ELISA, you know? So basically, we do it for many proteins, and we still continue with antibody to have an orthogonal validation. So we get plenty of information here. Recently, we went also we decided to use also sperm smears to check whether we could, get into deeper information. For example, yes, this ADAM20 protein, which is a known one, by the way. Others have no known function. And you see on sperm smears that the staining is really on the flagella of the protein. Just to give you a few words, ADAM20 belongs to the ADAM family it's a disintegrin enzyme, in fact, and in the mouse, knockouts on plenty of the ADAM members make that the male is sterile. There's no mobility of sperm. So you understand that probably those kind of proteins play a crucial role on the flagella. So you get, just simply with immunohistochemistry, you get clues on possible biological function. And then we have this huge consortium to get deeper into the understanding of what this protein can make. You select a candidate, you have to check whether this protein is conserved through species, mainly through up to the mouse, which is interesting, and you check whether you have an ortholog of the gene. That means that the protein is also expressed and has probably the same function in the mouse. You check whether there's a paralog. So what's a paralog? I guess you lost. A paralog is a protein that has nothing to do with it, but plays eventually the same function. If one is missing, the other one takes over. So you check there's no paralog, and then you contact a huge international consortium, which is the International Mouse Phenotype Consortium. They have 3 sites. One is in France, in Strasbourg, one in Australia, and one in the United States. Their work is to make a knockout mice for every single known gene. So you contact these people, and you see whether they already have prepared what we call the S cells. Maybe the mouse is already prepared, and maybe the mouse has already a phenotype. So through the screen on the internet, you can see, for example, here I have a candidate with a phenotype on sperm. Well, probably. So you order the mouse, they give you a couple, or 2 couples usually. You only pay for the transport of the animals, then you grow a colony and you study the phenotype, okay, and see whether you can understand how the protein works. But basically, it's roughly 25K for budget just for one single protein. So it can go pretty big and pretty fast and pretty expensive. So basically, looking at those missing proteins, just an estimation based on the mRNA expression on RA Express or RNA-seq Atlas, we expect at least 800 missing proteins to be discovered only in reproductive organs. And basically, roughly 650 in the testis, which is quite interesting. So basically, things you will see very often now is that people working on a specific subject, they tend to collect the whole bibliography of identified protein and make some sort of exhaustive repertoires. That's bullshit, okay? Because you have papers that have 20 years ago, you identified protein with ancient mass spectrometer with guidelines that were a little tweaky. And then now we have high-end mass spectrometers with strong guidelines. So the idea is to, if you want to merge data sets, even the old ones, you use the current guideline and you reanalyze everything, even if it was done 10 years ago. So that's the work of a friend of mine who is in Grenoble and who is a bioinformatician. So he takes all data sets, and another one to come very soon, and he's making what we decide to call the human sperm official build of the proteome, which is quite important to work. And now here I have work from an excellent person in my group who, on the basis of what we've identified, all these 3 recent papers on the sperm proteome, out of these over 300 proteins, 7 of these, the gene was not expressed in the cell line. How come it works? I mean, you see the protein, so the gene must be here, the mRNA. And in fact, these are RNA-seq data sets. This is transcriptomic, okay, of testis, epididymis, prostate, seminal vesicles. And for 7 of these, they come from the epididymis. The gene is in the epididymis. That means that these proteins are secreted by the epididymis during the sperm transit, and they will stick to the sperm. Let me give you an example. So we get to our friend at Human Protein Atlas, we get a good antibody. This is the mRNA expression. So you see, there's a lot in the testis, or maybe a little bit in seminal vesicles and adipose tissue, but well, not considered. But with the antibody, there's nothing in the testis, and this is the head of the epididymis. All these columnar cells whose role is to secrete protein, and you see the transiting sperm that's really highlighted for this protein, which is a defensin, by the way. So defensin are anti-infectious peptides. They have anti-infectious properties, so can be used for the sperm to defend itself during its transit through the female tracts. But also defensin has been shown in the mouse to be used for chemoattraction. So the idea is that maybe the sperm has a mean to be attracted by secreted protein by the ovocytes. So this is the idea here. So this is what we're going to work on over the next couple of years. So we have just recently designed a whole story, the whole study workflow, how we can get new knowledge on these 7 proteins. And at the same time with people in Sweden, so they have this huge millions of images on all the antibodies they're using every single organ. Go and visit HPA. It's a wonderful source of information. But over the years, I was looking at the testis information, and I say, well, this is really crap. This is not good. I mean, some people at the annotation, they will make they say, okay, it's expressed in spermatocytes. And you look at the section, you say, it's not expressed in the spermatocyte. It's expressed in spermatids. These guys are jerks. So I contacted them, and I say, well, you know this, anatomopathologists are not pretty good. I mean, but I can understand they don't know every single organ. So I say, well, we have a collaboration with people in India. It's not expensive, and they do a lot of work. Okay, say, well, if you guys design me an interface, I can spend, let's say, 5-10 minutes a day, and I will annotate a couple of proteins, and then we'll do next. So they design immediately under my recommendation. This is my control board. This is my panel, okay? When I go to HPA, and I have all, for each single cell, I can provide information that they can use, for example, say, okay, we have to ask and say people that they need to dilute antibody, for example. So we went to a testis deep annotation progress, and we made some kind of interesting discoveries, an interesting way of using the data. This is, for example, so we've recently annotated, I have recently annotated 500 proteins using the antibodies. Now, if you look at the mRNA expression in other organs, so each gray circle is one of the cells in the testis, and you see the colors here. The more it's expressed, the more color is deeper. The deep blue is the second organ that expresses the same protein. So, for example, here you see fallopian tubes. So fallopian tubes express 17 proteins that are in common with Pachytene spermatocytes, round spermatids. That's surprising. Or fallopian tubes, again, 77 proteins are common between round spermatids, elongated spermatid, and fallopian cells. How come? So this is the idea. How come from simple images you can get very interesting information? All of these proteins have known function, except one. What we can see here, they're expressed in the germ cell line. Some are expressed in late spermatids in the testis, or let's say future spermatozoa. Okay? This is fallopian tubes. And you have these very fancy cells in the fallopian tubes. They are columnar secretory cells. And on the top of these cells, you have cilia. And you have staining on the cilia, or in cells secreting towards the cilia. So here, for example, I have CFAP45, which is a protein with a known function. It's expressed in the cilia and in the flagella. And this one is a new one with unknown function. But you can see from the staining, it has a similar staining. So you go to one of the wonderful tools we have as biochemists, which is called gene ontology. It's a controlled vocabulary. So you have to be a computer geek to work with these things. And I have good geeks in my lab. Young kids are absolutely surprising sometimes. They don't know how to read papers, but they know how to fool around with their laptops and stuff. And in fact, with the gene ontology, we already know under the controlled vocabulary that this protein here is linked to cilia or flagella formation. So what do I say from my image? That protein with an unknown function has a similar expression pattern. It might be linked to cilia or flagella formation. This is the first layer of information on the possible role of this protein in the testis. So let's imagine that this protein is not properly expressed. Maybe it will not on the flagella, and maybe the sperm doesn't work. That's the idea here. You can also use differently these data sets here. So for example, it's another way of showing things. Between Pachytene spermatocytes and round spermatid, we have 68 proteins with expression, that they share expressions after meiosis. So you go to another wonderful tool, which is called Cytoscape. That's fantastic. For Cytoscape, every single node is a protein. You have the name. And the thicker is the line in between two things, the stronger is the interaction. It has been demonstrated several times. And you see all these proteins, the 68 proteins shared between these two cells. If you look at the gene ontology information, they already link to binding of sperm to zona pellucida, spermatid development, spermatid differentiation, cilium movement, spermiogenesis, acrosome vesicle, cilium, secretory granule. This is the whole story. Now, the limit now is your imagination. How come I could demonstrate that this protein is clearly involved in whatever acrosome vesicle in the sperm? So now it's the work of biochemists and biologists to work together and sit down on a bench and decide what to do to better express and better demonstrate what they want to demonstrate. So what we're doing at the moment, we're going deeper into the sperm proteome by subfractionating head, intermediate piece, and flagella. We're working on the study on those two interesting proteins. With our friend Thomas Fréhour in Nantes, we're working on the collection of sperm series from OATS sample, and we currently select candidates. There's two already in the pipe for knockouts on some of the proteins of interest. I will skip this one because I'm talking too much and I know I don't have time. So you know the story, fatty chip, for some of those guys here that are from France. We have this development ongoing in the lab. This is the 30 chip test to decide whether or not you should do a testis biopsy just by analyzing the sperm. You know the sperm cells in the testis, someplace in specific niche. So we're developing a triplex assay that should be ready by the end of the year. Next year, it will be packaging of assay kits, and our idea is at the end of next year to distribute to collaborative center for, I would say, 2-year period of validation into your daily practice. If you're interested, you can get to me and we can discuss further. And I will show you just very, very fancy thing at the moment. I'm proud to present this because I know we're the only one in the world to do this. The idea was, well, my obsession is say, okay, people are collecting sperm. And when I discuss with some people, I'm surprised because they say, well, epididymis, it's no use. It's not useful. I mean, come on. Nature doesn't make anything by chance. You know, epididymis is probably a very important organ in sperm maturation. So imagine you collect sperm by TZ, for example. No epididymis transit. How come you're sure that your sperm is perfect? Okay? You all know this problem. But I will address this problem using our tools. And we need advice for clinicians here. We need to sit down at the table and really discuss on what we're doing. So basically, I have to make a very short introduction on one absolutely fantastic technique, which is MALDI imaging mass spectrometry. It has been developed by a wonderful person named Richard Caprioli in Nashville. It's the way of using a mass spectrometer, not to analyze protein, but to analyze tissue section. So basically, Richard is working on the brain. You make a brain section, or whatever, testis section, but that's what we're doing in the lab. And you put this in a mass spectrometer, and it screens all the image, all the section, and at every single point, it will collect a spectrum here. And you reconstitute images. These are vitreal images of one protein that you want to follow, one signal. Okay, so what we've done is to do this on the epididymis. So on one side, we had the proteomics experiment, classical, and we were able to analyze caput, corpus, and coda. This is a rat model, obviously. So we have expression patterns of protein, and we know they evolve. And now we're using imaging mass spectrometry. This is quite fancy because we're not doing single images. We're doing the whole organ. So hundreds and hundreds of sections in a row. A rat epididymis is like a cotton tab, okay? A cotton tab is, well, 12 centimeters, and the whole data set is 3.5 gigabytes. It's absolutely huge, but we will work with it for 10 years. And we're here making a connection between proteomics and metabolomics, which is quite interesting here. So basically, we come up with a really high-end instrument. So this is a FTIC, or mass spectrometer, roughly 3 million euros negotiated price. So there's only 2 in France, one at Servier and one in my lab. Basically, this is a rat epididymis, and you have these exquisite images. The color here is just the expression of a metabolite. We can do it with protein indeed, and what we're doing, in fact, is we reconstructed the organ in 3D, and we can see the distribution of one specific metabolite, for example, here. And you see, okay, this one is expressed all along the tube, but this one is only expressed in a teeny section, anatomically different section of the organ. So 3D allows a very precise localization of molecular events. And in fact, let me give you an example. That's a beautiful example. This is a metabolite with a funny name, sulfogalactosyl ceramide. And as I said, you have this exquisite expression on one specific area of the head. So this is the head of the epididymis in the rat, and you have segments. So here we have an expression is segment 6, 5, 7, eventually 4. And when you look closer, so you have all these database. We love database in biology. And people spend time to get information into it. And you see this, these metabolites is in the sphingolipid metabolic pathway. So we get to the pathway because we know how to get there from another database. And this is it. This is the metabolite here. And this is the reaction, okay? And one enzyme that makes the reaction is called aryl sulfatase A. We have it, the protein, into our data sets. It's highly expressed in the caput, almost nothing in the corpus, and a lot in the coda. Okay, so we make the link between the two, okay? And then we look at the protein here, and what we know about this protein, its function in gene ontology. It says it's binding of sperm to zona pellucida. That's quite interesting. So we dig into the literature, get some old papers from 1990-something. The enzyme in the rat or the mouse is called ASA. So ASA, it's present in the surface of live sperm, okay? And there's a positive ASA staining in the head of live caudal, epididymal, and vas deferens sperm. That's quite interesting. So we get deeper into the story, and the idea is, okay, thanks to this metabolite, I was able to pinpoint ASA. ASA is acquired on the sperm surface during the transit in the epididymis. And we get again to HPA, get a nice antibody, and what we see, this is epididymis, and this is a pattern of secretion by the cells here. So it doesn't come from germ cells initially. So you see a lot of paper when you dig into the literature that people were almost approaching the role of this protein, as we know that that enzyme binds to MZP2 and MZP3 in the mouse. So, well, that's the protein that probably plays a key role in the interaction between the two gonads, two gametes. So then we get again to the International Phenotype Consortium, but to make the story short, the mouse is not ready yet. So we click on the button to say, okay, we have an interest here in France, the Britons, you know, Asterix village, and we'd like to get this mouse as soon as it's ready. Okay, I'm done. So this is, I hope I give you a flavor of what we could do with testicular proteomics. I mean, the idea is to use these tools. So you need guidance. You cannot imagine how difficult it is to drive a mass spectrometer, and especially to analyze the data it produces. But under guidance, I've learned this morning plenty of interesting projects, and I was taking notes that I have an answer for these people. I mean, we can help them to get deeper and deeper into the analysis of the problem. So I may stop and see you at lunch break to discuss further. And in fact, just to let you know, in the Human Proteome Project, I am launching with colleagues a very specific project, which is Testis Epididymis Proteome Project, with the idea to understand and unravel the mechanisms that underlie spermatogenesis and gamete maturation, and connect these to reproductive pathologies. So a special thank to all the people who participated to the work I presented today. I hope you're still alive and survive this extensive swim into fundamental biochemistry. And I would like to thank especially those people in blue that did most of the work and tremendous amount of work of what I've presented today. And I thank you for your attention. And also I'd like to thank my colleague Scott with the Bike Balls team. I always use this funny movie they have when I display things about the role of the testis and the interest we have of the testis. Okay, thank you for your attention.